So today I will be talking about our research on the development of ACTA sensor technologies for rapid virus detection in water. This research was initiated in 2015 as a Sensum Research Initiative project and has been carried out in my group in Japan. So the primary target in our sensor development is norovirus. Norovirus is the major cause of foodborne and waterborne gastroenteritis worldwide, and also known as an important viral contaminant in water. In fact, norovirus has been detected at a few points in Singapore's urban water cycle, specifically in untreated and treated wastewater and surface waters, according to the reports from Singapore. So these results have been obtained using polymerase chain reaction PCR-based methods, which are widely used for the detection of viruses in the environment. However, it takes at least a few hours to obtain the results and has limited on-site application. What we are proposing in this work is the use of a biosensor. A biosensor rapidly converts a biological response into a measurable signal. A biosensor consists of two major components, a recognition element and transducer. We use aptamer as a recognition element and electrochemistry or colorimetric measurement as a means of transduction. Now, what is aptamer? Aptamer is a synthetic functional DNA or RNA that falls into three-dimensional conformations binding to a specific target. This is an example of aptamer binding to norovirus protein. Aptamer offers many advantages as a recognition element of biosensor. First, its high binding affinity. The affinity is comparable to or even higher than antigen-antibody reaction. It is highly specific because it recognizes unique surface structure of the target. Its high thermal stability makes it robust against ambient temperature variation and reusable with regeneration by reversible heat denaturation. It can be produced via in vitro chemical synthesis, which provides high purity and constant quality with low production cost. Lastly, we can easily attach chemical modification to an aptamer for various purposes, including immobilization and labeling. So this is the framework of our research. First, we developed a MEMS-based electrochemical apta sensor. This was done as a Sensum Research Initiative project in 2015, as I mentioned in the beginning of this presentation. Then, after I moved to Japan, we've been carrying out the research on detailed electrochemical analysis of apta sensor performance and optimization. These two parts are on electrochemical apta sensor. The third part is on the application of colorimetric sensor using gold nanoparticles, which is also an ongoing research in collaboration with other researchers in Hokkaido University. We are still at the phase of proof of concept towards the development of these technologies for norovirus detection. So we used mouse norovirus as a model virus, and mouse norovirus specific DNA called AG3, DNA aptamer called AG3, reported in the previous research. So first, let me start talking about the development of a MEMS-based electrochemical apta sensor for norovirus detection, which was carried out in collaboration with Professor Jianmi Miao's group at Sensum and NTU. In the development of electrochemical sensor, we used a standard benchtop electrochemical workstation, as well as a portable electrochemical measurement system called WeStat. And the WeStat is as small as credit card and offered with a reasonable price, only around $130. We perform two types of voltammetric analysis: cyclic voltammetry, CV, for characterization of surface properties of electrode, and square wave voltammetry, SWB, for virus detection. For the development of Aptamer MEMS device, we immobilized thiolated aptamer on the gold electrode through the affinity between thiol and gold. These figures show CV profiles and before and after aptamer immobilization. As you can see here, aptamer immobilization on the gold electrode surface substantially reduced the peak current, which indicates that aptamer monolayer, self-assembled monolayer, was formed on the gold electrode surface. The VStat showed CV profiles similar to the benchtop workstation. Then I'm going to show you the result of virus detection by Apta sensor. This figure shows SWB profile, and this line shows SWB profile for a buffer without virus. When 500 PFU of virus was added into this buffer, we observed significant decrease in the current intensity due to the binding of the virus to the immobilized aptamer. Again, the VStat showed SWB profiles similar to the benchtop workstation. Here I summarize the key outcomes of our IRRI project. First, we fabricated novel MEMS aptamer device. We confirmed aptamer immobilization by electrochemical measurements as well as visual observation of labeled aptamer on the electrode surface. Second, we evaluated mini electrochemical system, VStat. By combining this MEMS aptamer device with VStat, we can potentially develop a portable Apta sensor platform. Lastly, we demonstrated potential ability for quantification because our experimental results showed clear tidal dependence response against virus inoculated in this system. So this result have been published and described in this paper already, published in last year. Next, so this is the ongoing research in my group at Hokkaido University. So we are working on detailed electrochemical analysis of sensor performance and optimization. We started from re-evaluating the conditions used in a previous study, reporting the first development of norovirus Apta sensor. Specifically, we evaluated redox species used as a probe in electrochemical measurement. We also evaluated electrode material, gold nanoparticle screen-printed carbon electrode used in this study. And we used screen-printed gold electrode. Lastly, we evaluated virus detectability of this sensor in pure water and tap water, and we determined response time. First, redox species, evaluation of redox species. We carried out electrochemical measurements with gold nanoparticle carbon electrode in 4 types of buffers. Electrolysis buffer without probe, without redox species, electrolysis buffer with ferricyanide or ruthenium only, or with both ferricyanide and ruthenium. This figure shows CV profile, and the color of the lines corresponds to the color of the text of buffer composition. These 2 peaks are derived from unique redox potential of iron and ruthenium ions. As you can see here, largest peak current was obtained from electrolysis buffer with ferricyanide only, indicated with blue line, which demonstrates that this buffer composition is the most suitable buffer composition. Additional advantage of not using ruthenium ion is that we can reduce the cost because ruthenium is a rare metal and expensive. Second, we evaluated electrode material using the buffer condition judged to be the most suitable in the previous slide. we used gold electrode, gold nanoparticle carbon electrode, as well as carbon electrode without modification as a reference, without and with aptamer. The color scheme is the same for SWB and this table. Again, we observed a clear peak here derived from iron ion. So we determined relative current intensity, which means the peak current intensity relative to the baseline, and compared different electrode materials. What we observed here is, first, we observed similar profiles from gold nanoparticle carbon electrode and non-modified carbon electrode, which means that the reaction on the gold nanoparticle carbon electrode, the reaction on the carbon material for gold nanoparticle carbon electrode is dominant for this electrode. Second, we observed highest peak current excuse me, from gold electrode after aptamer immobilization. So, so we judged that gold electrode is the most suitable gold electrode material. Then we assessed virus detectability using the conditions judged to be the most suitable in the last two slides, buffer probe condition and electrode material. So these figures show SWB profile in pure water and tap water with different amount of virus input. As you can see here, we observed reduced SWB peak current when virus was spiked in both pure water and tap water. And it was repeatedly observed that this response was observed within 1 minute. Here I summarize the key outcomes of the part 2. So we optimized conditions. Optimized conditions are for redox species, ferricyanide only. And for electrode material, we judged that a gold material, electrode was more suitable than gold nanoparticle carbon electrode, which was used in the previous study. And we determined the virus response time, and it was within 1 minute. The third part is on the application of a colorimetric aftersensor using gold nanoparticles to norovirus detection. The sensing mechanism contains aptamer and gold nanoparticles. In the absence of target, the aptamer is absorbed on gold nanoparticles, which maintains dispersion of gold nanoparticles, even at a high concentration of sodium chloride. The dispersed gold nanoparticles shows red color, whose absorbance peaks at 520 nanometers. However, in the presence of target, the aptamer binds to the target to form aptamer target complex, and aptamer cannot be absorbed on the gold nanoparticles. Bare gold nanoparticles tend to form aggregates, even at a high concentration of sodium chloride. So this changes color of the gold nanoparticles from red to blue, which decreases the absorbance at 520 nanometer and increases absorbance at 650 nanometers. Based on this principle, we recorded the absorbance ratio A650 over A120, 520, to indicate the aggregation degree of gold nanoparticles. This figure shows the absorbance spectra of gold nanoparticle in pure water with different amount of virus particles. In this figure, you can see only a subtle difference. However, if you plot the absorbance ratio against the virus input, you can see a clear tidal-dependent response. We observed similar response for artificially contaminated groundwater and filtered river water samples. So this is a summary of key outcomes of the third part. We evaluated GO nanoparticle-based colorimetric apta sensor as a novel virus detection method. We showed its applicability to virus detection, and our ongoing efforts include optimization of detection condition and parameters. And then we demonstrated potential applicability of the colorimetric apta sensor to environmental samples. In summary, we exploited electrochemical and colorimetric apta sensor technologies for rapid norovirus detection. And these technologies are simple enough to allow for on-site norovirus detection without specialized training. I would like to acknowledge the Sensum Research Initiative Program, which provided the opportunity to conceive this research and financial support to initiate this project. I also like to thank my colleagues from Smart Sensum, NTU and NUS, as well as Hokkaido University and National Institute for Materials Science in Japan. Thank you very much for your kind attention.
One question for Misaki. Oh, Stefan.
I'm Stefan Lutz from CERSI. Masaki, it looks like you have an excellent sensitivity for
Thank you.
norovirus. Do you think you need a concentration upstream, or are you just planning on passing hundreds of liters of water?
Yeah, eventually, I'd like to develop a method that can detect norovirus particles without concentration. But at the moment, I'm thinking of concentrating the norovirus first, concentrating norovirus particles first to increase the chance of detection using this sensor before we show the validity of this sensor for detection of indigenous norovirus particles in water samples. But in the future, yes, as you mentioned, we will try to develop a method that can detect virus without concentration in upstream. Thank you.